Good morning. Hello and welcome. I'm Janet Rossand, and I'm President and Scientific Director of the Gairdner Foundation. Thanks for joining us for our Gairdner Global Perspectives panel entitled One Health, One World, From Antibiotics to Zoonoses, presented by
the Gairdner Global Health Awards.
Before we begin, I'd like to acknowledge that the Gairdner Foundation is located in Toronto, Canada. We acknowledge that we are on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples. And today, Toronto is home to many diverse First Nations, Inuit, and Métis peoples. Today's event is part of Gairdner Science Week, which wouldn't be possible without our sponsors. I'd like to especially thank our Gold Gairdner Week sponsors, the University Health Network, York University, the University of Toronto, Sinai Health, Queen's University, London Drugs, and McGill University. I'd also like to thank our partners, the governments of Canada, Alberta, and Quebec, CIHR, and our media partner, the Globe and Mail. You can learn more about all of our sponsors by visiting the Expo booth section located on the left-hand side of your screen. And now I'd like to introduce our chair and moderator for today's panel, Dr. Stephen Hoffman. Stephen is Scientific Director at the CIHR Institute of Population and Public Health and Professor of Global Health, Law, and Political Science at York University. Welcome, Stephen.
Great. Thanks so much, Janet. Looking forward to this panel and chairing it. Excellent. So I have this honor of being your host for today, and just wanted to convey how delighted I am and how appreciative I am to the Gairdner Foundation for bringing us together today to talk about One Health, One World, from antibiotics to zoonoses. This is one of the defining issues that we are facing right now in our world. We're living through a pandemic where some of these issues have hit many of us in very real ways. But as we look towards the future, it's also clear that our health as humans is going to be increasingly interconnected with the health of animals and the environments in which we live, as well as with the health of people everywhere around the world. And so with that, the fact that we're coming here together, celebrating the awardees of the Gairdner prizes, but also to talk about these issues and think about what they mean for us today, also what they mean for us, what we need to do differently going forward, I think is quite an exciting thing. This is a 2-hour session. The first hour will be hearing from 3 world-leading presenters about One Health, One World, from antibiotics to zoonosis. First, we'll be hearing, I'll be introducing them in a second, but we'll be hearing from Peter Doherty, from Samira Mubareka, from Linfa Wang. And then in the second hour, we'll be going into a panel discussion. That's going to be an opportunity where me, as the moderator, I'll be able to ask questions. First of the 2 awardees of this year's 2021 John Dirks Canada Gairdner Global Health Awardees. So that's Malik Paris and Yi Guan. After which we'll be also joined on that panel with 2 additional world-leading experts, Amadou Saul and Sally Davies. So with that, what we'll do, though, is first go to the first part of this 2-hour session of hearing from the presentations. And first person I have the honor of introducing is Peter Doherty. Dr. Doherty, Peter is an Australian immunologist and pathologist who received the Gairdner International Award in 1986 and then the Nobel Prize for Physiology or Medicine in 1996, which indeed continues that tradition of the Gairdner Foundation and its selection committees identifying people who have made world-leading contributions in advance of the Nobel Prize committees, or foreshadowing what might come. Peter won the Nobel Prize for the discovery of how the body's immune system distinguishes virus-infected cells from normal cells. And today he's the laureate professor of microbiology and immunology at the Doherty Institute for Infection and Immunity, which was named after him. So we'll be now turning to Peter for his presentation. Thank you.
To be involved in celebrating the Gairdner Global Health Awards, to my friends Yi Yuan and Malik Paris from Hong Kong. And I've been asked to say something about the One Health perspective as background. The One Health perspective is basically a view that we need to address the connections between health and the environment, between life and the environment, life and us. And by doing that, we improve things for us, we accelerate biomedical research, enhance public health efficacy, expand scientific knowledge, and so forth. And it's a movement, One Health is a movement that's been gathering momentum over the last 10 to 20 years. In practice, One Health largely focuses on infectious disease and what we used to call comparative medicine. But if one attends One Health meetings, you can get a very different mix depending on the particular organizers, mainly the infectious disease perspective, but also One Health can go to a much broader range of topics, including the use of, say, plant agriculture to support animal and human systems. So it suffers a little from the fact that it maybe is too broad, but it does focus our thinking on the fact that we are part of the environment and we are part of the animal kingdom, which I think is enormously important from the scientific perspective. The comparative medicine part of One Health goes way back, because right from the beginning of modern science-based medicine, which really kicked off in Padua and Bologna in the 16th and the 17th century, and in the Royal Society of London and the Collège de France, we use animals, dissected animals, to try and tell us about how human beings work. Harvey, who worked out the circulation of the blood, studied eels and fish and birds as he established his principles. And Malpighi, as we know from the Malpighian tubules, also discovered the capillaries as he studied chick embryos and began the science of embryology. Then linking to infection and One Health, that actually precedes the discovery by Pasteur, or the work of Pasteur, that established, along with people like Robert Koch, the germ theory of infectious disease in the mid to late 19th century, because we had the principle of variolation and then vaccination when in 1796, Edward Jenner took lesions from the material on the teat of a cow and used that to vaccinate against smallpox, having noticed that milkmaids often did not become pockmarked. So that really is the first link between infection and human disease, in a sense. Louis Pasteur and Charles Chamberlain then, in 1879, developed the first scientifically based vaccine. Jenner's was a fortuitous discovery, but they grew fowl cholera. They'd learned how to grow bacteria. They grew fowl cholera. They went away for the summer, forgot about their cultures. When they came back, they didn't work anymore. And when they put it into birds, they found it didn't kill them, but it protected them against a subsequent challenge with live fowl cholera. That was our first vaccine priming virus infection challenge study. Later followed an anthrax vaccine, rabies vaccine, and so forth. The father of pathology, German Rudolf Virchow, very much focused on the link between human and animal health. William Osler, the great medical leader of the early part of the 20th century, also worked in veterinary pathology and scientific medicine and linked those two together. Then we have figures like veterinarian Jim Steele, who in 1947, after World War II, started the Veterinary Public Health Division of the CDC. So this was an emerging field. Then it was pulled together by Calvin Schwab in University of California, Davis, who pulled together the idea of one medicine. And we started to think in those terms. That thinking came much more, I think, from the veterinary than from the medical perspective. And in a way, it probably still does, though the recent experience of COVID may have sharpened those perspectives. Then in 2004, the Wildlife Conservation Society published the 12 Manhattan Principles that dealt with the movement of disease among humans, domestic animals, and wildlife. That gathered momentum, and in 2007, the One Health approach was recommended for pandemic preparedness at an influenza meeting in New Delhi with 11 countries and 29 international organizations. 2009-2012 saw the One Health approach recommended for pandemic preparedness, One World Health, USAID established emerging pandemics programs. CDC established the One Health Office, European Union and UN all came on board. So One Health really was an entity, and we started to have scientific meetings. I think I spoke at the first of the big infectious disease meetings. But here you see again that issue with One Health, with all these diagrams and little ovals and circles. It is an enormous span. And of course, if you're going to get traction with funding agencies, with politicians and so forth, you have to focus. And sometimes presenting this as an umbrella perspective can get in the way. And the most effective use, of course, has been in the infectious disease area. Comparative medicine still continues, and we can think of it as One Health. Here's, for instance, an example from Rob McCorkle at the University of Calgary, who looks at wound healing or is interested in wound healing. What he's really interested in is how reindeer regenerate their antlers. And basically, he's asking the question that if we look at how reindeers regenerate their antlers, which they do spectacularly every year or so, and you look at the reindeer velvet, then maybe we can get at the molecular principles from regenerative medicine. In actual fact, it's generally harder than it was to fund research through medical research agencies for comparative medicine, which I think means we haven't heard quite as much of it as we can. But there is some enlightened funding agencies who do that. So in the main, the agendas badged as One Health have dealt with viruses that jump from wildlife or domestic animals to spread within human populations. That initial incursion is zoonotic. It comes from animal species, but these are not zoonoses. Zoonoses are infections which are maintained in animals and that we generally catch from animals. These are infections that jump from wildlife or from domestic animal populations, as occurred in the 2009 influenza pandemic, into human beings and then become human diseases. They have an animal origin, but they are not spread broadly by animals. The principal viruses we've worried about for so long are those worked on by Yi Guan and Malik Pieris, the influenza A viruses, which are maintained in nature as infections of aquatic birds, jump readily into chickens, and of course the enormous expansion of chicken production in China and places like that has meant there's a greater risk, and then become established in us. This was pioneered by my colleague at St. Jude Children's Hospital, Rob Webster, who with his colleague Graham Laver first established the link between maintenance of influenza viruses in birds and the disease in humans. There are particular dangers with the influenza viruses, and we focus very much on the live bird markets. This is where people in cultures that like to buy animals live and then kill them for their own consumption are especially exposed to influenza viruses. And I expect you'll hear a lot more about that in the subsequent discussion. With the coronaviruses like COVID, we are concerned with the live animal markets. Again, a cultural feature of some societies where people buy live wildlife and use them for human consumption. And of course, we suspect that the SARS-CoV-2 virus, which is causing us so many problems at the moment, came across into humans from a live animal market, though of course this is contentious in the minds of some. We are constantly faced with viruses coming out of nature. Many of these are mosquito-borne viruses. Zika virus, we dealt with it several years back. It's an African origin virus spread by mosquitoes, been grumbling away in the Pacific. Suddenly it emerged in South America. And we had this problem with neonatal issues and the birth of children who had been infected in utero. That happens with many situations of sudden spread of pathogens from nature, including pet pathogens. They may be relatively quiescent for years, though they're in a particular country or place, but suddenly they'll break out and spread rapidly. We've had one great movie about it, and that's Contagion, which is very much parallels the COVID story, in fact. And if you watch that movie, which I think is worth doing, you can see some elements of what happened with COVID in that movie, including the recommendation of crazy therapies that basically were what Australians would at least describe as snake oil solutions. Now, we've been particularly focused on the viruses carried by fruit and insectivorous bats. And you'll hear much more about that, of course, as we go on in this symposium. The coronaviruses, we had 2 human coronaviruses circulating in people before the year 2000. Since 2000, 5 more coronaviruses jumped across into us. 2 new common cold coronaviruses and SARS, the first SARS virus, which burnt out, the second SARS virus, CoV-2, that we're still dealing with, and of course, the Middle Eastern virus that is still circulating, though at a low level. There are other viruses in bats, which, as you'll hear, have a particular organization, perhaps, of their immune systems that allows many viruses to be maintained in them. We can also be concerned about the Henipaviruses, Nipah in Southeast Asia, Hendra in Australia, and the filoviruses, of course, Marburg and Ebola, which I think it's concluded are pretty much likely of bat origin also. The coronavirus, we'll hear much more about now, and I won't expand on it in great detail. It's a standard coronavirus, and it's a intermediate virulence, much less virulence in the sense of killing people than either the original SARS virus or the Middle Eastern virus. But of course, it spreads so readily and it's gone everywhere, and it's a massive problem. Of course, there's also a massive opportunity if you're thinking of One Health in the sense of an opportunity for research that will really tell us a lot about disease in us. And it's a unique situation because for the first time ever, we've used a very sensitive PCR test to use as a widespread community screening mechanism. We've used it to trace contacts, and we've, as a result, soon learned that many people who are infected with this virus are relatively asymptomatic and would never present at a medical doctor or at a hospital. And we've also learned that many have mild infections in the same sense. But then we've also encountered this enormous problem of long COVID, which we can tie back to the infection because we have an original PCR diagnosis. And we've also seen the emergence of genomic epidemiology. And that, of course, was happening to some extent with influenza, but we've seen it happening very much at the local population level, where material from PCR has been sequenced, and we've been able, because of the minor mutations that happen in this virus, don't necessarily change anything that's serious, like virulence. We've been able to trace the particular virus variant that's causing an outbreak or a cluster of infection. So this has greatly empowered our capacity, or the capacity of public health officials and doctors, to trace what's actually happening in a community as a pandemic spreads. And what we're seeing, of course, is the first major pandemic in an era of transformed technology, which has basically changed much of what we can perceive. There have always been problems out there in medicine. We've thought for many years that viruses trigger later autoimmune disease. Now we will have that PCR diagnosis for COVID that we hopefully will keep the records properly and be able to link it to later development of autoimmune disease and perhaps see whether this is actually a trigger. And then, of course, there is the ill-defined clinical problem of chronic fatigue syndrome that physicians have struggled with for decades. And now we know an inducing trigger. And hopefully we can also pull that apart. Technological advances, what we've seen especially are the spectacularly rapid development and deployment of totally novel vaccines. These vaccines have been under development for decades. The mRNA vaccines had not been used previously in humans, were deployed very quickly and have proven enormously effective. Adenovirus vectored vaccines, again, a technology that had been around for a long time, had been deployed in Africa as an emergency use vaccine against Ebola, but never widespread in human populations. As we've put these out into millions and hundreds of millions, in some cases, of people, we've seen that there are side effects, that as we go forward, and if we can persist with these technologies, and it's undoubted we'll persist, I think, with the mRNA technology, will no doubt be sorted out. Some side effects, myocarditis, pericarditis, blood clotting, and so forth, that will be resolved. But they've shown the power of modern technology to go from a virus that was first isolated at the end of 2019, where the sequence was published in mid-January, to having fantastic vaccines out later in 2020. So this has been transforming. The other thing that's happening now, of course, is we're starting to see antiviral drugs come online. I totally underestimated the time that it takes to make and develop these things. Merck has a ribonucleoside analog, molnupiravir, that's going forward. Pfizer has an oral drug, a protease inhibitor, that's going forward. Now, the important thing about these antiviral drugs is they'll work against all the coronaviruses. Now, we have drugs already that work against all the influenza viruses. One of the problems with them is, though, that they're never given early enough. Now, if we use with influenza the approach we've used with COVID-2, that we do PCR tests on people immediately and then treat them immediately, we would do immensely better with influenza. And of course, if we develop those drugs for the other families of virus that could potentially be pandemic threats, and there's a major program from the National Institutes of Health, instituted by Tony Fauci, that will fund exactly that approach. We need these drugs because they could be our first use option if we have future pandemics. I think it's inevitable that we will have future pandemics if we continue on the path that we've been following. And the reason for that is the increasing prosperity in regions of the world that have live animal and live bird markets, and the increasing prosperity of a middle class, which is flying around the world and getting on planes. Unless we have very stringent screening mechanisms put in place, we're going to have more of this happen. We need drug stockpiles that can be deployed rapidly, and we need emergency response teams that could be dispatched to countries which do not have a well-resourced public health system. And the other thing we need is global agreements, total openness on the emergency of pandemic infections. And we need, as part of that, to be able to stop the planes flying out of regions where these things are happening quickly. We'd always operated in the flu mantra, and the flu mantra was, you can't stop influenza by stopping the planes. Well, countries like Australia and New Zealand, which are island states, did stop the initial Cov-2 by stopping the planes. It was as infectious as influenza. Many thought it was influenza initially. And even though we said it wasn't influenza, we approached it as though it was influenza. Now we know it's a very different infection, a much more complex infection than influenza. But until Delta came along, it was spreading with much the same infectivity as flu, and we did stop it. So you can stop these viruses if you stop the planes right at the beginning from the source of origin. There's a way to go, and there's, of course, we'll be thinking very much in terms of One Health, but I think that's a way forward. Thank you. It's a pleasure.
Great. Thank you very much, Peter. That was a wonderful presentation that really provides an overview of the range of issues that we're going to be talking about for the remainder of the session over the next hour and a half. I think from my perspective, Peter, there were a few key takeaways I think that are really important for everyone to make note of. One is that we live in a world that's characterized by One Health, by those interactions between humans and animals and our environment, and how that umbrella term can help reveal the extent to which our health is dependent on the health and those interactions that are happening in other domains, like the animal sector, in the environment. The second key message I heard was very much about that this challenge is getting more difficult, in the sense with more travel, with increasing trade, with increasing proximity between humans and animals. That's resulting in some of these threats increasing over time, something we need to think about. But also what was really emphasized there, which I think is quite hopeful, is the role that science has played in all this. And Peter's reference to the current pandemic, situating it here, really for me highlights the powerful role that science is going to play and must play to address these challenges, and that the kind of tools we have today are actually so different already than the tools that were available 10 years ago. Certainly those were better than the ones available 30 years ago and such. And then finally, though, I think Peter really highlighted how much of the agenda, though, is unfinished, and that if we continue with the status quo, we're not going to necessarily get to where we want to get to. So with that, though, I'm going to proceed right to our next presenter to talk a little bit more about viral transmission and aerobiology, to go in for focusing further, which I think makes very good sense in the context of a current pandemic, although this speaker who I'm introduced in a second has a particular focus on the influenza virus, which Peter mentioned quite a bit about. So Dr. Samira Mubareka, she's a virologist, a medical microbiologist, and infectious disease physician at Sunnybrook Health Sciences Center in Toronto, Canada. She's also an assistant professor of laboratory medicine and pathobiology at the University of Toronto. And since the outset of the pandemic in North America, Samira has been working on the SARS-CoV-2 virus, which is what causes COVID-19, with a focus on virus biology, bioaerosols, and exposure, as well as genomics and diagnostics. Samira, over to you. Thanks so much.
Stephen, and I'd like to thank Dr. Rassan, the Gairdner Foundation, for this very generous invitation. And I'd also like to extend my congratulations to the awardees, Malik Paris and Iguan. So I will help set the stage a little bit in terms of One Health by talking about our One Health approach to zoonotic virus transmission with a focus on bioaerosols. Really, the overarching goal of our research is to look at determinants of viral transmission. And we've been working in a number of different sectors: healthcare, agricultural, and sylvatic or wildlife settings. And I'll take you along this journey by touching a little bit on our work in each of these sectors. But before I do that, I really want to emphasize how important the real work is, the work that the undergraduate students, the techs, the graduate students, the postdocs, the RAs are all doing on this. Without them, we'd have no data, no analysis, no evidence. So thank you very much. So I don't think I need to explain to folks why it's important to understand modes of transmission for zoonotic viruses, or really for any viruses at all. But what I would like to emphasize here at the outset is really how complex and dynamic these processes are. Part of that is because there are both biological and physical factors that are involved, and it's important to study both of these, but also a number of different hosts and settings, and it will really vary between them. Viruses obviously have biological determinants for transmission and persistence in the environment. Different hosts have determinants for susceptibility and infectiousness. And also the environment plays a significant role. So factors like temperature and relative humidity, or the built environment, so ventilation and airflow, all play a role here. So how do we study bioaerosols? Dr. Doherty mentioned some advances in terms of tools, and I'm hoping that I can show you how we've been able to apply these. So obviously, if you're studying bioaerosols, you need a bioaerosol sampler. I have one example here. This is the National Institute of Occupational Safety and Health sampler, courtesy of Bill Lindsley, that many of us use. And you also need systems. So a lot of these are custom built. I have a couple of examples here. Number one, this is our artificial aerosol generation chamber. So we can aerosolize human pathogens like H1N1 and H3N2 seasonal influenza viruses to look at factors like humidity, as we did in this particular experiment, showing that at higher humidities, you're actually recovering less virus. Not that surprising. And then also we still use a comparative medicine approach. So we use ferrets for transmission experiments for influenza virus and other viruses. And again, another example here, we used ferrets to look at whether or not NSAIDs, non-steroidal anti-inflammatories, increased or affected viral shedding. A lot of people use these when they have influenza virus. And in fact, really, there's no difference whether animals are treated or not. And when you look at viral recovery from the air, the bioaerosols that they're emitting, marginal difference, but really not statistically significant. So these are a couple of tools that we use. We also, to get real life data, go to the bedside. And we started doing this with seasonal influenza virus some time ago, using these cassettes, or these filter cassettes, these low volume samplers, but also the NIOSH sampler I already showed you. And we've shown, although these are pretty small studies, we have been able to demonstrate that we can recover viral RNA both in the near range, which isn't surprising, so at 1 meter or less, but also a little bit further away from the patient, over 1 meter, over 2 meters, maybe even out into the corridor. And there's actually a pretty wide range of particle sizes that harbor viral RNA, but not surprisingly, the larger ones are recovered closer to the patient. Importantly, we also put these samplers on healthcare workers as they were looking after these patients. And again, small numbers, but we certainly had evidence that these viruses were making their way into the healthcare worker breathing zones. So when the pandemic hit, we were able to work very quickly on looking at SARS coronavirus exposure and risk in the acute care setting. One of the key questions that came up fairly early on was, what's the risk with high flow nasal oxygen? This is really occupational safety issue, but also a health access issue, because some jurisdictions have limited access to mechanical ventilation, and high flow nasal oxygen is an important option. But if it disperses virus broadly, then it's problematic. So we very quickly put together this breathing mannequin, affectionately known as Quarantino, and used a substitute virus, so a non-human pathogen called the bacteriophage, to look at how it dispersed in this breathing mannequin model in a critical care unit. And not surprisingly, we were able to show that with invasive ventilation, so that's a closed circuit, and this helmet with a PEEP valve, there was very little phage dispersion in the room. Not surprisingly, with high flow nasal oxygen, there was a fair amount of viral dispersion across all sites in the room. But we were very surprised to see that it actually wasn't that much more than if Quarantino was wearing nasal prongs, which a lot of patients use. So really not, at least according to this model, in excess risk. And again, in terms of access, we looked to see whether or not the same pattern reproduced itself on an air ambulance, not using phage this time, just saline and an optical particle counter, to determine whether or not there was more dispersion with high flow nasal oxygen than nasal prongs. And in fact, there really wasn't. So transporting patients under these conditions really seemed much more feasible in light of this data. And finally, in terms of the human work that we've been doing, we have sampled patients with SARS coronavirus who were acutely admitted and looked at their environments and contamination of their environments. So we looked at 78 patients and did surface swabs in their rooms, and also looked at the bioaerosols of 45 of these patients, and really recovered very, very little in the way of viral RNA from their bioaerosols and no infectious virus. We recovered quite a bit of viral RNA by PCR from surface swabs, but very few of these were actually yielded infectious virus, and it depended quite a bit on time from symptom onset. So earlier in the symptom onset, more likely to recover infectious virus from these surfaces. And we wanted to see, well, which patients are actually shedding and contaminating their environment. So not surprisingly, it was the patients who were hypoxic, who had high concentrations of RNA in their diagnostic swabs, who had comorbidities, and again, more recent onset of symptoms. And then last but not least, we wanted to have a look. We hypothesized that the viruses should be the same in the patient and their environment, but we showed that this was indeed the case by doing whole genome sequencing, and it was evident that patients were shedding virus into their environment that was practically identical to the one that they had in their respiratory tract. So moving quickly along to looking at agricultural settings and One Health, we started off looking at influenza virus of swine origin some years ago in collaboration with our colleagues at the University of Guelph and Ontario Veterinary College. And you can see that, it can vary from year to year. We know this virus is endemic in these populations here in Ontario. And also we were able to demonstrate that it was present in the air, depending on whether or not there was actually shedding from the pigs. And for that, we used oral fluids. It's sort of a nice way of getting an aggregate sample. They chew on these ropes, you squeeze it out, and then you know if the pen is positive or not. So we do tend to see contamination of surface samples and bioaerosols when oral fluids are positive. So that's not that surprising, but what other information could we actually glean from this? What other tools could we actually use? So we repeated this study and looked at whether or not we could get viral sequence out of the air and surface swabs. So we had a very high, this particular study, we had a very high rate of positivity, so very high viral burden in this case. And almost half of the surface swabs were indeed positive, again, not surprising. And then a quarter, give or take, of the bioaerosol samples were positive definitively. And then there were a number that were indeterminate, that may have actually just been low concentrations of viral RNA. The National Center for Foreign Animal Disease, Dr. Johannes Barhane, were very close collaborators on this, and they actually did virus isolation on a subset of samples. And I'm just pointing out the two air samples that were positive. One was a room level one, and again, we had personnel wearing personal samplers on their in their breathing zones, and certainly the virus, and this time it was viable, appeared to be reaching their breathing zones. Dr. Burhani and his team were able to derive whole genome sequences from these samples, both the environmental ones and the oral fluids, less so in the air samples, just because the concentration was a little bit lower. As you can see here, there's a strong correlation there. But we were able to get sequence out of pretty much all of to some degree out of most samples that were definitively positive. And this is important because it really helps with surveillance. You can do subtyping. You can even look at which clade is responsible for a given outbreak, for example. And then you could look at more hypothesis-driven questions like, is there a bottleneck? Is there a difference in sample diversity? In our case, we did not find that, but we did find diversity in segments of the viral genome, not surprisingly, mainly in HA and NA. So that's hemagglutinin and neuraminidase. So stemming from this is another study that Johannes led in Ghana with Osborne Quay and Tumasi, and we're just helping out here with the bioaerosol sampling during H5N1 activity in Ghana that was quite recent. And Tumasi looked at a number of live bird markets and backyard farms across the country, both doing cloacal and oral swabs on individuals, serology, and bioaerosol sampling over here. And not surprisingly, but unfortunately, 5% of poultry were positive. We were able to derive RNA from the aerosol samples in 4 out of 42 samples. And these not surprisingly, were H5N1, but also found H9N2. The analysis for this is ongoing. This is fairly new data. And then just very quickly, I'll try, Stephen, to catch up on time a little bit here. I'm going to speak very briefly about wild CoV, which doesn't really have to do with bioaerosols, but really important in terms of looking for SARS coronavirus to spill over in wildlife here in Canada. I'll really just be talking about the Ontario data. And luckily, there's not much to say in the sense that they've all been negative, but we have looked at raccoons, skunks, and minks. Some of these are live animal sampling. Some of these are convenience carcasses from trappers. Most of the samples did come from around Toronto, where there has been a high level of activity. But as the level of viral activity, and knocking on wood, abates, I think serology is actually going to be a much more important tool. Though we should still do PCR to make sure that there's no ongoing low-level circulation among mammals. We really don't need a mammalian reservoir. But we do know already that several wildlife species are permissive for and have been infected with SARS coronavirus too. So in summary, to conclude, there have been fantastic technical advances in aerovirology, and I didn't say this off the top, but most of the methods we're using are not really designed for viruses. There are some emerging methods that are being validated over the course of the pandemic, but really we don't have anything ready in hand yet that's been clearly optimized, but progress is being made. It's really important to look at both host and environmental samples at the human-animal interface. I didn't talk about wastewater at all, but that's another great use case for environmental sampling. It's really shown its value during this pandemic, and you can gain insights into pathogen characteristics and also exposure risk. And critically, all of this work was highly multidisciplinary. I think that is really the way forward in terms of bridging the knowledge gaps that we're currently plagued with. So I will stop there, and hopefully I'd like to thank everybody that I didn't have a chance to thank earlier on, and I'll stop at that point.
Great, Sam. Thanks so much for that fantastic presentation. I think perfectly came after Peter's, partially because Peter was talking a lot about transmission from animals to humans. You beautifully then helped us understand how that transmission can then continue in a clinical context from humans to humans through different means, but then also highlighting how some of the upstream surveillance work focused on animals, even though on one hand, some people might think it's quite distally related from humans, but actually that surveillance in animals is directly, that information is directly then informing clinical practice, which is, I think, just really helpful for understanding the complexities and connections here when we are taking that One Health lens to everything. And maybe the third takeaway message for me is I don't, at least not yet, have to worry about the raccoons in my neighborhood transmitting SARS-CoV-2. So that's good news for now. With that, let me thank you, Sam, and I'm going to now introduce our next speaker and our third of three speakers who will bring to completion this first section of our 2-hour session today, and that's Dr. Linfa Wang, who will be presenting about bats and coronaviruses and specifically what's special about this. And so Dr. Linfa Wang is a professor in the program in emerging infectious diseases at Duke NUS Medical School in Singapore. He is one of the world's leading experts in zoonotic disease, bat immunology, and pathogen discovery, serves on multiple World Health Organization committees, and has received numerous awards for his work on new and emerging diseases. Linfa's recent research contributions include developing antibody-based serological tests to detect the SARS-CoV-2 virus and the early successful culture of this virus from an infected patient sample. Linfa, over to you. Thank you very much.
Thank you very much, Steve, for the introduction, and thank the foundation for the invitation. So I'll be talking about bats and the coronavirus and what's special. I think, we don't need an introduction for coronavirus and the bats anymore, but I do want to, express my, heartiest congratulations for the two awardees today, and Professor Iguan and Professor Paris. So I thought an appropriate start point is actually to go through the papers that they published in 2005. So this is, the title is Identification of Novel Coronavirus in Bats, and published in, February 2005. And the sampling was done in July, earlier in 2004. So this is really just after SARS, and obviously everybody was looking for potential origins of the virus, and the bats was one of them. So it's very, very milestone paper. It's the first identification of coronavirus in bats. So I thought this is the way I pay respect to the two awardees. So I only have 15 minutes, so I'll go very fast to do a very brief review of emerging bat-borne zoonotic viruses. Coronavirus is only one of them, but the focus is to talk about bats and the coronavirus, and then why bats? Why bats are such good reservoir? And then to finish, really, along with the theme today, is One Health in Action. So my personal kind of experience working on the hanjovirus that Peter mentioned in Australia. So I started my career for virology in Australia, and then go all the way to COVID-19. So emerging bat-borne zoonotic viruses, basically 6 major outbreaks in 25 years. And personally, I was involved in all of them, but the 4 of them more heavily than the others. So if you look at the virus, right, 94, we had a hanjovirus in Australia, followed by 98, 99, Nipah in Malaysia, Singapore, SARS started from Guangzhou, 2002, 2003, MERS, Middle East, Saudi Arabia, 2012, and then 2014, West Africa, we had Ebola, and then back to China, Wuhan, 2019, we have COVID-19. So exact 25 years, and we have 6 major outbreaks. Some are highly deadly, and some are very transmissible. And if you look at the world map, right, it starts from Australia, went to Malaysia, and then Guangzhou, Middle East, West Africa, back in Wuhan. So I think it's clear that no place is safe, right? And as Peter Doughty says, it's not an if, but it's a when the next pandemic will happen. So bats and the coronaviruses, so as I said, 2005, the two awardees of today, they published the first paper to identify that. But that's a big year, 2005. After that, if you search for bats and the viruses, you look at the publications per year, it really shows up. Last year, over 600 in the 25, and this year is still climbing up. Okay. And in 2005, after the paper that Yi and Maliki published, that 2 other papers came, a little bit half year later in October. So one in PNS from the Hong Kong U group, and one that I led, it's an international consortium with 5 institutes coming from China, USA, and Australia. So basically, this paper identify bats as the reservoir of SARS-like viruses. And from this research, I was involved with the WHO kind of committee, and that's how I made the interactions with the 2 awardees. And since then, we become really close colleagues. So I was serving on the scientific research advisory committee that Malik was on. So that's how I met with Malik. And since then, we have been exchanging information, collaborating, and with Yi, so we both joined this first mission to China to investigate the reservoir, the origin of SARS-CoV-1. Now we can call SARS-1 virus, and early transmission events. So in comparison, 18 years ago, the mission to China was much less controversial and it's much more scientifically based than the second mission early this year to investigate the origin of SARS-CoV-2. Nevertheless, I think we had a very productive time. So these are the 8 international experts invited. The baby is actually transformed into E. So E was not there when we took photo, but these are the 8 international experts. We eventually led to the discovery of bats as the origin of SARS-CoV-1. So in the One Health kind of arena, when you have a pandemic, thinking of where the virus come from is quite important scientifically. And but I think we have to differentiate between the different viruses. The outbreak virus is the one that caused the human outbreak and pandemic. But we believe that this virus is relatively new. Otherwise, it would be already detected. And so it's evolved from that progenitor virus, and then the progenitor virus is in turn evolved from ancestral virus. So I think it's beyond any doubt, that the coronavirus we have discovered, which caused the zoonotic outbreak in the last 20 years, for SARS-1, MERS to SARS-2, the ancestral virus, we believe all from bats, So the human outbreak was 2002, 2012, and 2019. And then the reservoir host for the ancestral was defined or identified 2 years after the original outbreak for SARS-CoV-1 and 1 to 2 years after for MERS-CoV, but 5 days after the human sequence was obtained, because internationally, a lot of group and the Chinese group already have this sequence in their freezer, basically. What we are really searching for now is what we call potentially an intermediate host, the animal X. We still believe that most likely there's an animal X involved in transmitting the virus to human, the progenitor virus. Due to the, obviously lockdowns and travel restriction, international collaboration is difficult, but we still managed to have a collaboration with Dr. Supapan's group in Thailand. From one sampling event, one cave in Thailand, we discovered both molecular and serologic evidence of SARS-CoV-2 related virus circulating in bats and in pangolins in the region Southeast Asia. Recently, a preprint was posted, and it's a much more close kind of a relative has been discovered in bats from Laos. And interestingly, I think it just makes 2 points that first of all, if you look at the coronavirus genome, they were able to, if you divide into 15 segments, if you use each different segments, phylogenetically, they're positioned differently. So that indicates, that just like we know already, coronavirus can recombine very easily. Another very important is that if you use the segment that encoding the key functional domain, we call the receptor binding domain of the spike protein, which make interaction with the human ACE2 receptor, then you can see that, on top of these are the human viruses, and then these 3 new bat viruses discovered in the bats in Laos are very close to the human viruses, much closer than the red G13, which was in the scientific domain and the intermediate topped a lot. So now we have 3 viruses very, very close. And the key discovery from these studies is that now we have a bat SARS-related coronavirus with an almost identical receptor binding domain of the SARS-CoV-2. And more than that, we know that although we detect a lot of coronavirus in bats, live virus isolation is difficult, but they manage to isolate live virus with only 2 amino changes in the IBD. The live virus can infect different human cells, can be neutralized by COVID-19 sera, and the virus, as I said, is a product of the multiple recombination events. So now, again, year and a half after the Wuhan sort of outbreak, and with travel restriction, with all the hindrance to do field research, and yet, we still discover so many SARS COVID-2 related coronavirus all the way from Japan to East China, Southern China, now Laos, Thailand, and Cambodia. So that, it's a direct span of at least 5,000 kilometers. And we know that because the bats of that genus, we call the rhinoceros bats, they are widely distributed from east coast of Australia all the way to where I am now, Southeast Asia, Southern China, Middle East, Europe, and sub-Saharan Africa. So I think in theory that this type of virus can emerge any of these locations. So why bats? I don't have time to go into the talk today, but just to really alert people, if you don't know too much about this flying mammal, it's amazing. During flight, their metabolic rate can go 15 to 30-fold of their resting rate, and their heartbeat can go around 1,000 or over beats per minute. And the same bats during hibernation only beat 6 beats per minute. And their body temperature during flight can go to 38, 42 degrees C, and that's not due to any disease, it's just their normal physiology. So what we believe, I have been studying, bat immunology for the last 15 years, and I don't have time to go through the data, but suffice to say, there's a lot lesson to learn from bats, as a very, very unique reservoir of viruses. But obviously, bats did not evolve to harbor virus. The ability to harbor virus without disease, I think, is a kind of side byproduct for their 65 million years of adaptation to really, adapt to flight, because the stress I already, illustrated during flight. And the end results is they have a much better defense versus tolerance balance. I mean, whether you're a human, mouse, or bat, we all have our defense system. If the defense system is not working hard enough, then you get to disease. But if they go overboard, you get disease as well. So the balance is very important, and we believe that in bats, they do that better than us. They have certain genes, for example, the heat shock proteins, the autophagy genes. They have a high base level, so they're basically battle ready, because they need that, because they fly every night, and during flight there's a lot of stress they need to deal with. On the other hand, they have a mechanism to dampen certain overreaction that made them, can carry virus without disease and can coexist. That's why they're such a good reservoir. But anyway, I don't have time to go into it. I mean, you can read all the papers, So I will finish really my talk with One Health in action. So that's kind of my personal experience of how to really apply One Health in dealing with this emerging bat, zoonotic viruses. And start with Australia. That's how I, started my infectious disease career. We discovered that bats as the natural reservoir of the hantavirus, and that use horse as an intermediate host. Since that discovery, we have discovered Nipah virus in Malaysia and Singapore, Nipah virus in Bangladesh and India, and now Nipah virus in the Philippines. So they can use intermediate host horse and the pigs to transmit to human, or in I think India and Bangladesh, we believe it's a direct back to human transmissions through date palm juice. And in terms of the get a One Health approach, is that in Australia, there was a huge demand to try to ask scientists to come up with a smart way to prevent that disease transmission. So we discussed about, to develop a vaccine for bats or develop a vaccine for human. Neither is really that feasible. So we decided to go for a vaccine against horses, but really to killing two birds with one stone is to protect the horses, but also more importantly, is prevent a transmission to human. So that vaccine was developed in a very short time period because it considered as like emergency approval. And it's almost now 9 years ago that to the date that was approved for release. So this is the first licensed vaccine in those days and in today against a biosafety level 4 agent. Okay. And since the vaccine was applied to horses, we have not had a single human case in Australia. And then now I think the international community, especially people working with the MERS coronavirus, I think you use the same strategy to develop vaccines against camels so that to prevent that in the future MERS outbreaks. So I will finish really to discuss the challenge now we're having with COVID-19 and SARS-CoV-2. In the previous talk, Sam already did some surveillance in the wildlife in Canada, but now, more than a dozen species has been proved that can get an infection. When you talk about zoonotic diseases, you think of spillover as an animal to human transmission, but for COVID-19 now we know spillback can happen. So we have another term called forward zoonotic transmission and reverse zoonotic transmission. So this is a new challenge because that with so many human beings infected all over the world, the chance of pass the virus to animals is a real danger. And traditionally, we think of wildlife, think of this as a natural reservoir, intermediate host, or amplified host. But now for SARS-CoV-2, they can also serve as feedback host. And then if they're successful to maintain that, then you get this, what you call a new sort of unnatural natural reservoir. So this is my hypothesis, although the origin of the virus has not been resolved, but I have a strong belief that the ancestral virus is from a rhinoceros bats, most likely in Asia. And then they transmit to animal X. As I said, we still don't know. And then to human, followed by massive human to human transmission, and it's still ongoing as we speak, right? And plenty of evidence. The most kind of high profile human to animal transmission was the mink farms in Europe to start with. And the virus mutates and transmits back to human. That's bad, but not the worst scenario, because at least these are farm animals, and the farm animals we can control relatively easily. What we're worried about is that they can now pass to new kind of wildlife. Bats in America, for example, they don't have the runoff bats. The natural reservoir is not there. But from studying the ACE2 receptor in bats, we identified there are many bats, actually susceptible. So it's not impossible for humans to pass this virus to bats. And we know bats, due to their unique immune system, will not display disease. So that will be an issue, that then the virus will mutate, and then they transmit to animals X, Y, Z, and then they come back, become, human SARS-3 and 4. So with that, I'm going to finish my talk to say that One Health is definitely the way to go. And, we need to work close in order to prevent the next pandemic. And with that, I'd like to thank you for your attention.
Linfa, thanks so much for your presentation. A bit of a scary ending there, but providing the perfect segue into our panel discussion. Certainly, I think, Linfa, some of the key messages from my perspective are really around how viruses evolve and how it works, going from ancestral virus to progenitor to the viruses that we see today that affect in humans. But then also the different role that natural reservoirs or intermediate hosts and spillback hosts that you talked about, how that all ends up affecting, well, in the case of the current pandemic, the daily lives for people everywhere around the world. So a big thank you for that, and perfectly setting up for what we'll do next for the next hour, which is going into our panel discussion. And for that, I'm really delighted and really honored to be welcoming now 4 exceptional leaders who'll join us now for discussion about One Health and what we need to do about it. And the first 2 we'll introduce are Dr. Malik Paris and Dr. Yi Guan, who are the 2021 John Dirks Canada Gairdner Global Health Awardees. You've heard quite a lot about them during over the last hour. This event is very much a celebration of their work and many contributions that they have done throughout their careers and are still doing for advancing all of our health. And specifically, they were awarded the John Dirks Canada Gairdner Global Health Award for their contributions to understanding the origins and options for control of newly emerging infectious disease outbreaks in Asia. And so Malik is a professor and chair in virology at the University of Hong Kong. And Yi is chair professor in emerging viral diseases, and the Daniel C.K. Yu Endowed Professor in Virology, also at the University of Hong Kong. In addition to both, really delighted and honored to be joined by 2 additional world leaders. First, let me introduce you to Professor Dame Sally Davies, who is the 40th Master of Trinity College at the University of Cambridge in the UK. And she currently is as well the UK government special envoy on antimicrobial resistance, about which, Sally, I know we have talked lots about, and we will talk a lot about over the next hour. And as well, Sally served as the former chief medical officer for England and the former chief medical advisor to the UK government. Also delighted to introduce Dr. Amadou Sall, who's the head of the Arboviruses and Viral Hemorrhagic Fever Unit, director of WHO Collaborating Center, and director of the Institut Pasteur de Dakar in Senegal, which is part of the Institut Pasteur International Network. And Amadou's research focuses primarily on diagnostics, ecology, and evolution of arboviruses and viral hemorrhagic fevers. Welcome, everyone. Thank you very much for joining us today. I expect this is going to be a great panel. We're going to talk about the science of One Health. We're going to talk about how this affects people. We're going to talk about how do we get greater action to address the kind of scary threats that I think Linfa just presented on, but also we heard from Peter Doherty earlier, as well as from Samira Mubaraka before, after that. And so let me give the first question to one of our two awardees. Malik, we heard quite stark assessments of the threats that are posed by ignoring One Health. Can I first turn it to you, building on your research? What's your assessment of that threat? How much should we be worried? How much do we need to be acting on this today? Over to you.
Yes, so I think we heard both from Peter and from Linfa about the continuing threats that we face from of pathogens, particularly viruses, and of course antimicrobial resistance problems jumping from animals to humans. And I think, over the last 2 decades, we as a species, we have dodged a number of bullets. We had SARS in 2003. Of course, it came, as Linfa explained, from bats through civets and to humans, but that was contained. Then we had the 2009 pandemic of influenza, which spread worldwide. It could not be contained, but it was very mild. So we seem to, and then we had Ebola, but it was extremely severe, but it could be contained because of its modes of transmission. So all of these came from animals to humans. We had COVID, and that's the bullet that hit. But I think, we cannot, even as bad as COVID is, we cannot assume that COVID is the worst pandemic that we may face. It is certainly possible that even more virulent pathogens can come from, from animals to humans. And we really have to use this One Health holistic approach to try to risk assess and address these threats.
Malik, a quick follow-up. A lot of people around the 100-year anniversary of the Spanish influenza pandemic had talked about a once in 100-year type of pandemic. Is it, in light of what you're seeing, in light of your research, in light of sort of what you described there as near misses, I think is how you framed it, is that paradigm of thinking about once in 100 years, is that the right paradigm, or has that actually maybe been misleading us to accidentally underestimate the kind of threats that these potential zoonotic viruses and other aspects can cause and lead to?
No, I mean, I don't think we can assume that we, can relax for 100 years before we face a similar threat as COVID. And this is, I think, both Peter and Linfa explained the reasons for it. It is basically human behavior, what we are doing, whether it be our encroachment into wildlife habitats, our large-scale the way that we do livestock in large scale, but also moving livestock from, thousands of miles from one place to another. And then air travel. So these are all changes in our human activities that are increasing the risks of, first and foremost, these pathogens jumping species. But then once they do, spread extremely rapidly worldwide. So I think the threats are much more accelerated at this present point in time.
Great. Yi, I'd love to turn it to you to get a sense of the assessment of this threat from your perspective as well. One of the things that Peter mentioned in his initial presentation was he made reference to the movie Contagion, which I think many of us have seen, maybe all of us have seen. Certainly I will always be, I always require my students to be watching that as required watching in advance of the first lecture, because it gives, from my perspective, it gives a very good sense of some of the things that will come. Of course, though, now we don't have to watch the movie. We're sort of living through it in many different respects. And so, Yi, I'd love to get a sense from you, is this threat getting worse? Because a lot of the things we're facing today seem to have been things that you and others have been talking about will be coming, and we're now living through it. Yi, over to you.
Me?
Yi, over
Sorry.
to you, yes. About your assessments of the threats and whether it's getting worse.
Yeah. Is my microphone open?
It is, yes, we can hear you.
Okay. I just missing the first part of the question, but about to see the pandemicity of the virus, if we based on the last 2 decades, experience, and also if we go further, go to the last century, go to even Spanish flu. If based on we understand, if we're using the influenza A, flu A, it seems that just in last century, every 10 or 20 years, there still have some virus spread over to the human being and cause the pandemic. So our initial experience and the knowledge experience accumulated, saying that every 1 or 2 decades, we have one pandemic. But for the coronavirus pandemic, this COVID-19 is the first one. Actually, we don't have any preparedness. Even the ecology of this virus in nature, or with how this virus develop into the human-to-human transmissible, there are 1,000 questions piled up. And at this moment, a lot of political atmosphere don't allow the scientists quietly and rationally discuss how this virus emerged. Okay, so this is certainly will be stop us for next pandemic preparedness. Especially we got to understand there are more than 1,000 species of bats in nature. Okay, and the Linva's talk gave the example. Those examples is majorly those bat species like a flu bats that working closely within the biosystem across human being. Okay, like a SARS and the COVID-19, this type of virus, if they stay in the natural reservoir, it's saying the very rare to cause the so mature virus in human directly. We need some amplified machine to speed up, make the virus is much more efficiently transmissible between human to human. Yeah. So this is the question. We never forget that. Even in the virus slipped in the bats, different type of bats, we found the virus actually is a major replicator in their, in the intestine system. Okay, not in the respiratory system. So we have working in the year 2007. It's tried to defy many different type of coronavirus. They are hosted in different species of bats. What we found in consensus. First, all the virus residued in those bats seemed older than the virus we found in our domestic animal. Second, those viruses have all very complicated evolution history that keep going on different recombination, sorry. Okay. If we pile up all the evidence we know at this moment, actually, if any human infection case directly from a bat without any intermediate host, they only happened symptomatic case. Okay. We can never say our humans metabolism system with different species of bats should just share the same. If that is the case, I think every month we have a pandemic.
Yeah.
This is we cannot avoid the key question. Yes, one health, one world, but actually for bats it has a better world. For our human being have our other world. Okay, we got to think in developed way. Certainly, is human behavior directly interfering? Okay, like those farmed wildlife in China. Actually, before the outbreak of COVID-19, I visited the Zhejiang Delta region of southern China. Those wildlife for carry SARS to the human being, all the species come back. Okay, in the animal market. So that's the question: whether we come back this time or maybe next year. Okay, I stop here.
Oh, but that's that's both very helpful, Yi, but also quite scary. So we will go to, I promise everyone who's participating and watching this right now, we will move to maybe more positive things, but it's really important to note that the depth of the challenge and so much of this challenge is not always talked about and among public, and it's sometimes probably too easy to just not address it because it's complex. So this is really helpful. Amadou, I want to go to you next. So just staying with pandemics for a second, because we're all living through one. We keep on hearing, whether it's from Peter, from Malik, from Yi, about how the increasing sort of human-animal interactions and existence of animal reservoirs and such are they're actually increasing over time in the threat that's posed, but also that increased threat then requires changes in the way we respond to these epidemics or pandemics. And you're a world leader in mounting and thinking about responses to epidemics. Would love to get a sense from you, to what extent are those greater connections between people around the world and how humans are living closer to animals? How does that inform the kind of responses that we need to these things when they inevitably arise?
Yeah, thank you very much, Stephen, and thanks for having me. And I'll take this opportunity also to congratulate Malik for these awards. This is really a great proud for our institutions and network, and we've known Malik for years, and this is very important. Now, coming back to your questions, what we really, I want to mention a couple of different things about this changing environment and how it affects response. I think there is very key important element from this One Health that we need really to have in mind. I think number one is we need really to make sure in terms of preparedness, we have the right people and defending the right questions. So I think the part of building the right capacity in terms of workforce is something that has been very much impacted by the nature of the epidemic we're facing now in the context of One Health. What I mean by that is when we're talking about today an epidemic, let's say for Ebola or SARS and things, we are talking about something where we have at the same time people very much specialized in wildlife, with people very much specialized in virology, working in a completely different environment, and in a process which is impacted by mobility, by different sort of knowledge. And if you have all these different people to work through a multidisciplinary approach in the very different contexts, culturally and in terms of movement of people, it's very important that the people handle that, have this sense how to get about all this ecosystem. So training the people in a way that they can seize this sort of diversity and complexity is, for me, one very important step, where the capacity building is something that we need really to make sure when we train people, whether it's a student or a health professional, that they understand we are living in a context where we have all these moving parts linked to the fact that we're talking about the One Health approach. The second thing, which is critically important for me in terms of response, is that we need to make sure that for the response, we have as much as early as possible the right information that can trigger some right action when it comes to response. I'm just going to give the example today of the SARS-CoV-2. I mean, unlike other epidemics, the virus was detected. China published the sequence, and very quickly people start designing diagnostic tools and surveillance being set up. And this has been quite unique and remarkable. So the point I'm trying to say that having an early detection system based on cutting edge technology to detect or to know the sequence is something that is really important to inform the response and make sure that it is appropriate. And in this regard, we've been very good in making sure that when it comes to human, we have the information. But I think we have a lot to do, making sure that we can seize the information which is from the animal context. We're doing quite well when it comes to domestic, but when it comes to wildlife, it's a little bit more complicated. For instance, what we're trying to build right now in Africa is that through catching mosquitoes that are carrying a lot of these emerging viruses, where you're talking about Zika, dengue, and all this, we can have a system where we combine really the power of the entomology going to the field and sampling, at the same time sequencing from the field and getting the information very quickly into some sort of data sharing approach. Yeah. So having this signal as early as possible would help a lot actually to do the response. And the last comment I wanted to make, besides capacity building and setting the system in a way that we have early response, particularly with getting edge technology, I think is also all the part where capacity need to be built in terms of having the right medical countermeasure to respond. Today, I mean, if you look at what is happening, one point concerning vaccine or diagnostic have been really very, very scarce tools at the very beginning. And if you talk about Africa, we can witness now that in SARS-CoV-2, once again, to take that example, vaccine is not at the level where it's supposed to be. And this is particularly linked to the fact that the ecosystem and the vaccine manufacturing is not there. So I think to get your question, these three important elements I think are important. Build the right workforce. Second, make sure we get detection early with getting detection. And third, make sure we have the ecosystem to manufacture and deploy actually the medical countermeasure.
Yeah, Amadou, thank you. And one of the things that stuck out with what you just said is how important it will be to actually not just collect this information and data and insights, but actually make use of it, bringing it together so it can inform actions. And for that, Sally, I want to turn to you. I'm going to ask you a question to help us talk about antimicrobial resistance in a second. But first, if I can ask you to put on sort of your former chief medical officer hat for a second. Building on what Amadou just said about that, the need to be able to integrate these insights in ways that are then actionable and can inform government responses. I wanted to get a sense from you about maybe if you can help us peek behind how that actually works in at least a government context. Because when Peter Doherty was presenting about One Health, he had this figure that highlighted the full range of issues that come up in a One Health context. And when Amadou was just speaking about making use of that data, it kind of overwhelmed me. How does a chief medical officer of a government around the world, a chief scientist, how do you bring that together to act on it? Sally, please.
Thank you. Let me start by thanking the Gairdner for an interesting debate and discussion, and congratulations to Malik and Yi for winning the prize. It's difficult because at every interface, whether it's about One Health or ambulatory care versus hospital care, there's a problem with sharing data, with getting it across that interface and actually understanding it. And we talk about One Health, and we all know what we mean is what happens there impacts what happens there, and we humans are at the center of it. And we haven't mentioned environment yet, but actually we can't do everything One Health. So the question is, how do you get each group doing a really good job and highlighting when there's something that needs to be shared? And how do you get those interfaces that work on trust? And Yi was talking about preparation, and so was Amadou. And we did quite a lot of desktop exercises so that people knew what we thought might be different, and who wanted to know it, and how could you share it. Oh, and by the way, you might share telephone numbers and emails, just so that if the formal route didn't work, can you do it informally? So there's all of that building that we need to do. But then there is something about modern digital technology. How can we do that to make it much easier and everything? And I think we've made leaps and bounds through this pandemic, but there's still further to go about integrating data. There are concerns about privacy preservation for individuals, but in practice, most of it is. It's not about Sally Davis in London or Cambridge. It's about patients and people moving around and what their data shows, and how bringing it together you can get so much more out of it. But we aren't where we need to be yet. It's not smooth. It will always have interfaces that are difficult, but we need to find ways through. And we need the scientists to be part of it. It can't just be the policy people talking to each other. If it is, then it becomes too complicated and doesn't work.
Sally, I want to stay with you because for those who've been tuned in for the last hour and 25 minutes, they might accidentally have the impression that One Health is only about pandemics. Now, of course, we're living through one, so it meant that we had to be talking about One Health in that context. I want to get your help to talk about antimicrobial resistance. Even just simplifying antimicrobial resistance to its death toll, it's just staggering. I mean, already, at least some estimates point to 700,000 deaths each year, but by 2050, the expectation that annual deaths could be up to 10 million people each year, which I note would be actually double what the COVID-19 pandemic has caused over the first 22 months, because we are actually at that point where tragically we are just about to hit the 5 million death mark for the current pandemic. So that's a double pandemic each year is what this thing could become. Sally, can you share with everyone listening in, what is antimicrobial resistance? Why does it keep you up at night?
I would also add that, of course, it's affecting low and middle income countries rather more than it is affecting the rich countries. So what's happening is Darwin's natural selection. It is that when infective organisms, we're talking mainly about bacteria, but it happens with viruses, fungi, parasites, all infective organisms can spontaneously develop resistance to treatments. And then they can multiply and cause disease, illness, and even death. And what happens is that you get resistance developing to antibiotics, antivirals, antifungals, which makes these bacteria into superbugs. And over the years, we've been able to open the cupboard and get out a new treatment. But now we're running out of treatments. There are occasionally people who die of gonorrhea, even in America, where they've tried 23 antibiotics and they haven't worked. Across the world, we're seeing in India, for instance, about 60,000 babies dying every year of sepsis, of which most have untreatable sepsis. So because of resistance, we've seen a very, very unpleasant breakout of typhoid that's drug resistance in Hyderabad going up to Lahore, which has meant that the death rate in children has gone from about 4% to 40%. And we've only got on top of that one with a vaccine. So this is very real, but it's got many manifestations. I've been telling you what happens in humans when we get resistance. But of course, we share infective organisms with animals. And so resistance can develop in animals if they are treated with antibiotics. So while we would all see the importance to the food chain and to happiness of treating sick animals, did you know that more than 70% of antibiotics are used in the food chain not to treat sick animals, but actually to promote growth, and because it's much cheaper than cleanliness and sanitation to prevent infections? So clearly, resistance can occur in animals, chickens, pigs, beef, that can transmit both in the abattoirs, both as cooking and through into humans. We also have the problem that more than 70% of antibiotics are excreted either in urine or feces by animals into the environment. And it's very slow to decay. So we're getting a build-up in the environment that can cause resistance to occur in bacteria and viruses and everything that are free flowing in the environment. Meanwhile, most of our antibiotics are made in India and China, and as yet they haven't found a way to ensure that the discharge from factories is at safe levels, and from many factories it is massively high and very toxic in our waterways, going into rivers and then into seas and oceans. So it's a multifaceted One Health problem, and all of us have to work together and individually in our areas.
Thanks so much, Sally. Malik, if I can go back to you, building on what Sally just said. I mean, both Sally and Amadou, and before that, Yi offered a bit of an agenda for action that's needed based on, based on some of your work, based on the work of thousands of others who've contributed to better understanding this space. What would be your sort of prescription for what needs to change in light of this One Health lens that we're discussing today? What would be some of the either policy actions or practices that you just think we're not doing right now and we need to do in the future?
Yes, I think there are two aspects to this. I mean, what we need to do using One Health, and on the other hand, how we can enhance One Health. So these are two different things. But first and foremost, in regard to what we should be doing in a One Health context, I think, as was clear in the presentations and discussions, these threats to human health, and indeed the reverse zoonosis that goes back from humans to animals and the environment as well, is continuing to take place. And we need better surveillance so that we can have a horizon scanning of the potential threats out there. Now, that is not sufficient, of course. You also need to be able to risk assess what you find. Because if you look, just taking viruses as an example, look for viruses in wildlife or domestic animals, you'll literally find thousands of them. Which ones do you take seriously for countermeasure development? And this is where I think in the influenza space, we have come a significant way forward, especially after the last 2009 pandemic. So there is in the influenza community, what is called with the US CDC developed what is called IRAT, Influenza Risk Assessment Tool, and the WHO has a similar thing called TIPRA, Tool for Pandemic Risk Assessment, where it is not just the detecting a virus, it's not just the sequence, but characterizing its biological properties and making a risk assessment as to which ones are important. So that is one important thing. The other thing is understanding the common modalities, pathways by which these viruses emerge to humans. So, for example, in the emergence of zoonotic influenza, I think the work by E and myself have shown some of the common pathways, how the viruses evolve, and also how they amplify, for example, in these live poultry markets. And you can take fairly generic measures where you reduce the risk, irrespective of which particular virus might be posing a threat. So understanding the pathways of emergence is important as well. Yeah, I mean, I think that there's a lot that we need to do, and we need to do that in a One Health context.
Great. You know, when you describe that, Malik, it sounds so sort of simple and obvious that we just need to do that, right? Like, it just sounds so rational. Of course, though, it's difficult. And I guess for that, I'd love to then, with the time that we have left over the next sort of 20 minutes, love to better understand how do we actually get to a place where we're able to act on what seems so obvious and the need so clear. And so for that, Yi, I'd love to first go to you. You've been someone who's been talking about the need to address wet markets and other challenges to prevent zoonotic disease. Why is it that it's been so difficult, from your perspective, for governments to act on what seems so obvious?
Okay. You know, the virus emerging or invade human being. As a symbolic case, that happened each and every day, okay, in different corners of the world. But majority, these interspecies transmission events are dead end. That means they only kill or cause sick of those infected patient individual. They are not cause any trouble at the population level. So we deal the two level things. One is we call the symbolic infection case. Another is large scale outbreak of human disease. Actually, this is like the two-stage things. This is what we have done in the flu. Okay, we know at the very beginning, all the human flu are zoonoses. They come from animal, birds, even other mammals. Actually, we are also not so how the virus really mature and developability. I mentioned largest the last 2 weeks, more than 2 in every week. Then we understand how this virus, this lineage, H5, H9, they step by step get involved. Yes, but we are brave as a cry wolf because this virus not persistently to human infections. They only occasionally cause the pandemic, human case. So I think this kind of long-term effort surveillance in the field to tracing after virus evolution, I think let us have the ability to avoid some early stage of interspecies transfer to human. Now coming back to this COVID-19, why we failed on this occasion? Because the previous work not be accessible by anybody else. It's very few people working on this site. Okay, now if we mention from the Hong Kong 1997 bird flu incident, we change year by year. Eventually, we remove the human infection case of all bird flu virus infection in Hong Kong. Just a few miles away in the southern China, okay, we in the last 2 decades have 4 lineage of virus emerging here, and each of the lineage potentially can infect human, but the pandemic didn't happen in this region. I think this is quite automatically thinking. This is the long-term effort by me and Malik, our large team, persistent effort. But for other virus group like coronavirus, because they host so diversity, the distribution and the back migration is really, really hard. Whether we consulted like surveillance program.
Oh no, I think Yi, if you can hear me, I think we've lost you. Okay. You can't hear you up, and I think we've lost you now. But we certainly heard from you that one of the challenges is very much around that there are so many events that could be that next pandemic event. So if there's 200 potential events every year, and if you're a government or a prime minister, and you need to figure out which of the 200 do I act on, and maybe the right answer would be none. It can become really hard. Pointing to Malik, your earlier points about the need for those not only the data, but then also the assessments capability, the risk assessments, and then Amadou, your earlier points about the capacity needed and the people and the data and the systems to make that happen. Amadou, can I ask you to build on what you mentioned? Why is it so difficult, in addition to the fact that it's hard to find out which is the event that requires sort of action, what else might, from your perspective where you sit, would be contributing to the difficulty of doing what seems so obvious that we need to do?
Yeah, thank you very much. And I think this is a great question if you're looking forward. I see three main difficulties and challenge. I think number one is really about governance. We do not have the right governance to address this question at the global level. Even though we've done tremendous efforts and have great achievement with international health regulations, and we're building different sort of mechanisms with WHO, IE, and this is still not yet there in terms of making sure that we build this infrastructure of trust where people feel comfortable about sharing information and be in a position where actually people can trust each other and do the work together. Because we see definitely with that COVID or other disease, if everybody join forces, we can have a vaccine within a year, something like that. So I think really having the right governance where we can take the good decisions and move forward the plan is something that is missing. I think number two is I feel like we need to work completely different, and we haven't found that way. I'm not talking about governance, but really to be innovative and creative into building this collective intelligence. Let's say if you talk about surveillance today, like Mali was rightly actually addressing it, we would need actually to go to wastewater. We would need to do surveillance by collecting data from mobility, putting all that together and build this collective intelligence around surveillance and not having numbers and detecting virus, but look at all the complexities and have a fine way to move forward. This is something that is really lacking. Being smart together, I think we haven't found the right thing besides having sort of framework to work together. And I think the last thing, which is really for me lasting, is that we as a scientists most of the time have been very good in doing excellency in science, but not really good in linking with community and build what people call now as scientific citizenship, meaning that going to the community, talking to them, build solution together, listening to them, learning from them, and build things. This connection today is something very complicated for which we have not been very good. And I think this is really a major obstacle because even though with science we can build fantastic vaccine, unless we can convey the message and avoid vaccine hesitancy, this may not be very helpful. So I think working on these three things, governance, building collective intelligence, and connecting to our community, we may actually do much better into handling pandemics. Over to you.
Great, thank you. And I see Yi, we have you back, which is great. We built on your comments, and Amadou very helpfully highlighted some additional challenges for facing why it is that we face inaction, even though it seems so obvious. But Yi, did you want to finish your earlier remark so that everyone can benefit from it? Back to you.
Yes. What I mentioned is about when we evaluate the one virus pandemicity, this is very difficult. This is the limitation of our knowledge. Even for flu, okay, we have 50 years working history on this virus. We're still not so clear what's the major determinant, okay, of the gene to cause the pandemic, to trick the pandemicity, okay. So the only way to solutions is for pandemic influenza preparedness is we prepare vaccine, okay, pre-pandemic vaccine. We have prepared diagnostic reagents, even a drug. But for that case, if we put this scenario, go to the coronavirus, pandemic coronavirus, is so difficult, okay, because the coronavirus behavior is so hard to define, okay. Even we have a very good lab, Okay, even especially after this event, we have some conflict or argument about the source of this virus and the behavior of the virus. Why so mature for human being? Okay, so this is got great trouble for many people who are long term working in this field, including myself. Okay, and so these things is yes, what we should do and what we shouldn't do. If we do, we alarm, we raise our alert about this threat from the animal. Okay, with the wildlife and the domesticate virus animal. We'll be praying that you guys are crime wolf. Okay, to threaten the world. Okay, but when really things happened, okay, people try to minimize you guys' effort. Then this is the real world we're facing in each every day. Okay, how difficult we maintained, our largest surveillance system in southern China for 2 decades. Okay, then we isolated more than 140,000 virus in the stop. This is all the work we prepare just for one virus, okay, through the pandemic. But for other, I don't think, I think the surveillance is definitely, and the communication to set up the international community about global surveillance, okay.
Yeah, to be able
I
to
think
actually
give the, at least give the people to have a chance to alarm, to make an alarm on, not an alarm off. So this is we need to call different government to do so.
Yeah, I think that's so important. I wanted to then build on that. So Sally, if I can go back to you. So Yi talking about those international mechanisms to sound the alarm. Amadou also bringing on the table about governance. You're a thought leader on how to be organizing global architecture around antimicrobial resistance. You're actually part of that architecture too, in many respects, serving as a member of the UN's Global Leaders Group on Antimicrobial Resistance and other mechanisms. Sally, how do we build the kind of governance structures that Amadou was talking about? We hear it in the context of the pandemic treaty. We hear about it in so many ways, but you've been thinking about it for quite some time now on antimicrobial resistance. What works? What doesn't work? How do we go? Where do we go from here? Thanks, Sally.
Thank you. And let me build first on what Yi was saying. What we need to think about is insurance, and that's at 3 levels. It's about governments. I don't know a single government that doesn't want a really effective fire service to put out fires the minute they start, or if they have a sea or an ocean on their edge, how they can do a rescue service for things when sailors come adrift. So they need to think about putting in enough money so that we'd never get in this position again. And that does mean investing in public health, in data, in science. But then we need civil society insurance, because we were, let me speak for Britain, our inequalities through our obesity and poor health and poor housing of people who were in poverty played out and gave us a much worse outcome than we really did expect, and that's civil society insurance. And then, of course, and this will then lead into AMR and global governments, the private sector has a role to play. They have a role to play in giving us obesity. They have a role to play in the data collection. Malik and Yi and Amadou have been talking about movement, well, expenditure data, restaurant entry data, that sort of data. Terribly important, let alone, of course, developing the better ways of disinfecting, better ways of preventing infections, better ways of treating them, and better vaccines. So when you come to AMR, we have all the complexity of a pandemic, because AMR is a pandemic that's slowly growing, but people haven't seen the salience, because despite the death figures that you've been describing, people don't feel it. We hope that because of COVID, they will, and they'll suddenly realize, we're trying very hard to make them realize, that spending on prevention and getting it right is much cheaper for the economy and life saved and everything. And because this is a One Health thing, you can't just leave it to the WHO. They are central because it's human health we're worried about. But I'm worried about animal health because that's our food chain. So the FAO has to be there and the OIE. I'm worried about the environment because we get distribution of AMR and development in the environment. So we need UNEP. So somehow we've got to bring all of them together. I personally have argued for some years we need at the UN not just a Security Council and an Economic and Social Council, we need a Health Security Council that could then ensure that each of those are doing the right thing, because no one can take it away from those governance structures. It's how do we coordinate them and help them. I think we need a treaty or a proper legal instrument. And if we have a pandemic one, then the pandemic of AMR must be in there. I worry that we shouldn't reopen in international health regulations and that it will be very difficult to get a pandemic treaty. So I think we need to, and you're expert at this, Stephen, but we've worked together at the Leeds Castle meeting and things, look at what instruments have worked. One of the things we need is fewer antibiotics using. So how do we use a mechanism like the Montreal Protocol, sort of mechanism for getting everyone to reduce their use, inappropriate use? Of course, part of development is using them more when people are sick. How can we stimulate innovation of vaccines, better prevention and new drugs? And we're working through the finance ministers of G7 to try and get agreement that each G7 country will try a new method of innovation pull, delinked in general. And then we can learn from those, meanwhile using the same buckets of how you value antibiotics. After all, we have to value them not just for the good they do to an individual, but the good they do to that health system, that society, and the good they do just sat on the shelf over there, ready to use if we need it. So we're going to have different elements, but I still think we need something overarching to make sure that all the players play their role, and we may need something like the Montreal Protocol. And finally, I'd say that we do need to have the evidence brought together, and we are waiting and have been waiting far too long for the promised the independent panel for evidence for action in AMR. I want to see the panel, and I want to see that it's truly independent. I worry that it may be captured by one of the UN agencies, and IPCC is not. We need something to happen that's independent. Over to you.
Great, thanks so much, Sally. And I will give then the last sort of question. Madhu, it'll have to be. Hopefully you can help me answer it quickly. It's Sally talked about a lot of, I thought, quite exciting things that seem to be coming up to try. It's focused on one issue of antimicrobial resistance, but the kind of playbook for reforming governance to address a broader range of issues, I think is quite a helpful one. And when I think, as someone who focuses on global governance in my own research, I think a lot about, when I think of one of the most exciting developments, I actually often think of the African CDC, so the African Center for Disease Control. I just see, but I'm from where I sit in Toronto, Canada, I see it as a very exciting initiative. Wondered if you, Amadou, have any thoughts on that. I know the African CDC, for example, recently hosted a big African One Health conference, taking on that perspective, but any thoughts that you have about that newer entity and its promise as a final comment for this panel would be helpful. Thanks, Amadou.
Yes, thank you very much, and for giving me this opportunity to really emphasize the fact that the role of Africa CDC has been amazing in this. And the power of this came really from the fact that it has been a very good mechanism to unite expertise, political will, and translate that into very action that are very focused on delivering what is needed there. The very important aspect of that has been that if you look at Africa CDC as part of this pandemic, what they've done initially was get a very strong mandate in terms of governance from the head of state, which is the highest level of authority you can have, to say, okay, let's build a continental strategy. And let's address first through different initiative testing, that was the pact, and also all other different initiatives, which were and up to now, to take care of things like vaccines or surveillance or different aspects. And I think this is critically important. And I think the fact that this initiative has been taken to bring together the governance piece and to bring together the expert piece, which is the second very important aspect, where each of the different institutions was supported to either lead or document and support some of the technical aspect has been really, really important. And the last part of what Africa CDC is doing is it is strengthening through different organization, the whole ecosystem of Africa when it comes to preparedness. And I think for this reason, it has been extremely powerful. And I think really it's an opportunity to give a great tribute to the leader of Africa CDC, which is John Kengathon, who did amazing work and building that to the level and make all of us as Africans very proud, but as citizens of the world, very, very happy how Africa behaved during this pandemic.
Thank you, Amadou. And with that, let me congratulate again Malik and Yi for the award, and thank you for your service and the work that you've done over the last decades, which, as we see from this panel discussion, has been extremely important from a scientific perspective, but also actually already very much improving people's lives around the world. So a big congratulations to you. And Sally, Amadou, thank you so much for joining our awardees in this panel. This was an amazing conversation, much appreciated. With that, then, let me turn the floor over to Janet Rassant, who is the President and Scientific Director of the Gairdner Foundation, for final remarks. Janet, thank you.
Yes, well, thank you, Stephen, and thanks to all our speakers for really what was quite an extraordinary panel. And thanks to you, the audience, for joining us today. Now, this is not the end of Gairdner Week by any means. Up next at 12 p.m. Eastern, we have a symposium on the future of cancer precision medicine. And I invite you all to come back and watch. Tomorrow, we have our laureate lectures, followed by our gala celebration, a virtual gala, where you will see each of the 2021 Gairdner awardees receive their medals and give their acceptance speeches. Trust me, you don't want to miss it. So thank you again.